So welcome everyone. I see that the clock is 11 CEST and I think 10 in the morning in UK. So welcome you all to this Structure Meets Function Fragment-Screen seminar conducted by Instruct-ERIC. I just want to, my name is Sridhar, working at the Goethe University Frankfurt and mostly into the fragment screening by NMR. And here today is Instruct-ERIC. It's the door for structural biology research infrastructure where people can apply to various research facilities. And this could involve research visits, training, internships, and R&D funding, what Instruct would then also assist with. Within this Instruct, what we are conducting right now is the Fragment-Screen as a complete project. And Fragment-Screen, as many of you would already know, is that in the protein world, you have a surface, and the protein surface has pockets in them, wherein the ligands can be designed in the pockets. And one of the majorly and nicely accepted ways of designing a ligand is to start with a very small molecule, means 100 Daltons chemical molecule, and then you grow this one to 450 or 500 and make it like a drug molecule. And the path is not so easy, as I said, but it has many challenges. And in order to address certain challenges, Fragment-Screen is coming together, to bring instrumentation, computational methodologies, and workflows into one framework. Yeah. And in this, we then try to involve several technologies like what you see as X-ray, NMR, cryo-EM, mass. But one of the major things is also data curation and data distribution. And this is one topic what we would address from Genevieve, who is here. And the second one important is currently artificial intelligence is being utilized in a powerful way, and how we can use this one in order to design better fragments. And this would then be covered by Jannis, who is here. So the first talk, I would like to introduce Genevieve, who is here from EMBL-EBI. I happened to listen to a very nice talk, and in Oxford recently, Genevieve has her, she's a PDBe annotator at the EBI, and she has very many interests. And one of the things is she's also a trained protein biochemist, and protein purification is also one of her specialties. So not only the PDBe annotation, but she is very well known to this one. Second is, second talk would be the language models, which means use of artificial intelligence. This would then be by Jannis, who is working for IBM Research in Zurich, in Switzerland. He has trained himself as really during his PhD in language models for molecular design, and his main interest for developing AI for science or so. So this is the main interest. So I now leave the stage for Genevieve to get going with her presentation. Thank you.
So today I'm going to talk to you about a subtype of structures that get deposited to the protein data bank, which are structures where fragments are bound that have been determined as part of screening a fragment library. And in my talk, I'm going to start by talking about data structure more generally, the data structure utilized by the Worldwide Protein Data Bank, and then I'm going to go into how we're leveraging this or building the capacity to leverage this to capture more information related to fragment library screening experiments. And we're doing this not only by building an extension to our data structure, but actually having an additional file type called investigation or investigation MMSIF files. I'm also going to briefly do an overview of fragment screening research, touching on some foundational concepts, and in particular, the commonality and diversity in this type of data. And an important aspect is that we've built code to help us, a software application to help us generate these files. And I'm going to finish by talking about this, how we often consider structures in the context of other structures. And so some of the broader implications talking about capturing this metadata for fragment screening also relate to capturing metadata for sets of structures for other methods. So for example, time-resolved crystallography data sets, there's often multiple structures determined, and there's potentially a defined relationship between these structures that could benefit from an investigation MMSIF file. So starting talking about data structure. So potentially, well, most of you probably have encountered this kind of page. So this is if you've published an experimentally determined structure, you've likely encountered this. This is the page that once you've entered your methodology, you'll see this page. And at the top, just to give you a hint, if you ever get lost a little bit of what you requested, there's a request ID here that corresponds to, if on this case, the request was for an X-ray crystallography structure, so just a PDBe ID is requested. But if you've indicated that your method is NMR, you'll have a BMRB ID might be listed up here as requested. If you've got a cryo-EM structure, there might be EMDB ID up here requested. So this is potentially a nice place to sometimes look if you're trying to orientate yourself. And so a key thing that perhaps is not so obvious when you're depositing a structure is you upload your coordinates in MMSIF. And this is an example here of an MMSIF coordinate file that's been uploaded. This corresponds to a small structure, just one chain. And when you upload it, it has about 3,000 lines. And as you fill in all of the forms in the deposition interface, and then you get to the point, finally, you've got all of the information filled in, you click the green submit button, all of a sudden that file size actually jumps because all of the information you've written into the form is now embedded into the MMSIF coordinate file. And then as it gets processed by an annotator like me, we do various additions to improve annotation, to improve validation, even more lines are being added to this file. And by the end, this relatively small file has actually doubled in terms of its size of lines as we've added all sorts of additional information in a very defined way in this file. And what this means practically, if you're ever in a situation where for some reason the hold expired is about to end and you're not ready for your structure to become released, if you take the file that we've sent you along with your validation report and reload it into a new deposition interface, almost all of the forms in a deposition interface will automatically fill because it's now all of that information is defined in this file that we've sent you, the MMSIF coordinate file along with your validation report. So that's a handy tip for you in terms of depositing structures. And so I asked DALL-E AI to create an image for what, to visualize what an MMSIF file looks like. And it created a couple of images, but I really like this one because I think it really highlights that essentially an MMSIF coordinate file is a set of chunks of data, lists and tables and other lists and other tables with defined relationships between them in this sort of complicated looking cube shape here. And all of this information is defined in a publicly available dictionary. And so you can find out what the relationships are between all of this information. This is the way that I typically will view these files, which is a combination of a freely available software called Visual Studio Code and an extension for SIF. And essentially what's nice about this particular combination in terms of viewing the text of that file is that when you mouse over, so this is the atomic coordinates, and if you mouse over each of the columns, you can find out which each column corresponds to. And so here I've highlighted that if you mouse over this one, you can find out that, oh, it corresponds to the occupancy for this particular line, this particular row in the table. And so I kind of think about this as being the atom site table, and then each of these items corresponds to headers of the columns. And if you take this file and view it in any of these visualization softwares, then it just looks like it's just a normal coordinate file. But underneath, this is sort of what it looks like. And these, the coordinate file, they have essentially, as I say, it's this network of tables and lists. So you'll have your atomic coordinate file here, maybe. If you visualize it here, it has connections to another list and another table over here. And in the case of this, I'm showing some examples of extra information or additional information related to what is it? Is it a protein? Is it DNA? What's its name? Is it chemicals? What are their names? Is it DNA and RNA? What is this, or protein? What's the sequence over here? So it's this really interacting sort of set of tables and lists. And why is this useful to us? Well, the data structure that we have enables us to have sanity checks to make sure that the correct information is being captured, sensible information is being captured, and also we have controlled, we have controlled vocabulary to so that we don't have any typos creep into the database. Another way in which it's helpful is obviously if you're trying to find a cohort within the database that corresponds to a particular interest you have. So here I've just done rapid assessment of which coordinates are determined using cryo-electron microscopy. You could add additional terms defining what kind of resolution those structures were, and so you can define a particular data cohort. We also have MSF categories that provide links to other databases so you can find related information. So in this case, I'm highlighting Empire for cryo-EM, diffraction image data, links for X-ray. And these all get listed in this particular category here. And if you notice, this is the MMSIF dictionary that I indicated before. All of this sort of information, how it's structured, is defined here. And again, another thing that's really useful about this data structure is if a new method we're told scientists contact us and say there's a new method that we're using. In this case, recently, scientists contacted us and said there's a new tomography method that we're using that's called 4D STEM. And we added that to our dictionary so that now that's an option that can be selected. And so what I've sort of touched on as I've gone through so far is the various aspects in which this particular data structure is so useful to us, which is the fact that we can standardize it, we can extend it to cope with new methodology. And obviously linked to this is the fact that we can automate it. We can build tools related to data management because we've got such a defined structure. And one of the aspects related to this is something called FAIR, which you may or may not have heard of. So FAIR is data management, and I tend to think of it as just good data stewardship. It's how do we handle our data in a way that makes the data still findable, makes it accessible, makes it reusable, and this term, which is a little bit of a mouthful, interoperable, so that it potentially has the most capacity to be used in different purposes. So interoperability is the idea that even processes that we haven't envisioned yet will be able to use this data because we have it so clearly and well defined that new applications can use that data for something that we haven't yet envisioned. And that really comes down to the fact that we have this publicly available data dictionary that defines all of the information, how it relates, and what should be there. So with fragment screening, what we've been working on for a while now is to essentially develop an extension to the dictionary, which captures more information specifically about fragment screening experiments. And this is the schematic laying out how those different tables and lists interact for a fragment screening MMSIF file. So just touching, we've sort of talked about this, it's already indicated in the intro. Why do you do fragment screening? So the motivation for fragment screening lies in the capacity, the ability to sample chemical space. And this can be thought of sort of from two angles. One is thinking about it in terms of the protein surface. So if you think about proteins, they have these extremely complicated surfaces that small molecules might be able to find either a surface or a pocket where something could bind. And if you shrink the probe size, then essentially you reduce the complexity of that protein surface. And this is nicely, eloquently discussed in this paper, 20 years on the impact of fragments on drug discovery. So that's thinking about it from the protein surface. So I kind of often envision it like a climbing wall, a blind man trying to climb a wall and trying to find footholds or handholds. And having that smaller probe just means that there's more possibilities for finding that pocket. Another reason to do fragment screening is coming from the chemical perspective. So essentially chemical space is really vast. So here I'm showing sphere sizes with logarithmic representations for the number of chemicals. And so Zinc 15 and the CAS registry database are two databases that really describe chemical space in terms of chemicals that have been synthesized or discovered and published. Or slash are purchasable. And so we're talking 200 million chemicals, and that's not even thinking about the chemicals that you can imagine to be synthesized, which that space is even vaster. And so if you imagine trying to screen all of these chemicals against a target, this is just would not be possible in a wet lab. EU OpenScreen has developed a library where they've got chemicals from across Europe, and they've got about 100,000 chemicals. And by making lower molecular weight chemical sort of looking at the larger molecular weight chemicals, so in this case, 350 gram per mole chemicals, and then looking at fragments that might be found in multiple chemicals, they've been able to sort of describe their relatively diverse chemical library in a subset of fragments, so 1,000 fragments, which is obviously, it's a lot easier to screen 1,000 chemicals than it is to screen 100,000 chemicals. And of course, another reason to do fragment screening is because it works. And increasingly, there are drugs coming into clinical trial, which have been developed from a fragment. So here we've got an example with erdafitinib, where you've got a small fragment that was the original hit, and then it gets developed over a number of years into a larger chemical that eventually goes through the various stages of clinical trials and comes 13 years later to market to treat malignant cancerous tumors, in particular bladder cancer is one that gets one of the cancers that gets treated with this particular drug. And this is from a blog that you can from Practical Fragments, where they've got a nice summary of the chemicals that the drugs that are coming to market that have had a fragment hit as a starting point. So fragment screening, there's a number of different methods that can be used. There's a number of biophysical methods: ITC, DLS, DSC, MST, SPR, mass spectrometry. And then obviously there are ligand-based NMR methods that can be used. And in addition, there's atomic level structure characterization. So the structures that get deposited to the PDBe, where a whole structure is determined with a fragment bound. And these there's a variety of methods that could be used in order to determine these structures. In terms of thinking about X-ray crystallography and the methodology that's being used there, often what's happening is there's a crystal plate, and the crystal plate is having various chemicals, various fragments, being essentially using sound to drive those chemicals onto the drop and soaking those fragments into the crystal and then collecting that data. And there's various beamlines that have been streamlined to do this. And one of the things we've noticed at the PDBe is that sometimes these structures will be deposited, but they won't necessarily end up with a publication. And this is, I've got this image here of Achilles with an Achilles heel. So one of the things with doing a fragment screen rather than a small molecule screen is you're likely to get a hit, but it might be challenging to evolve that hit into a more potent target, which is what the second presentation today is going to cover. Talk more into that space. So in terms of fragment screening experiments, they're quite diverse methods that can be used, but they all share a commonality in that there's fragments involved, there's a target that's being hit, there is fragments that are both hits and misses, there's multiple negative results, multiple data sets, multiple files, and multiple supporting data files. And if we break it down even further, thinking about the kinds of information you might want to capture when you do these kind of experiments, we want to capture what the fragments were. We want to capture the experimental conditions, the sample preparation, the type of the target that was used. Obviously, things like who the authors were, the dates, facilities, all of that stuff, and links to other databases where various bits of information might be. And so just highlighting here are some different colors. So there's information that sort of more generally could apply to not just fragment screening experiments, but other experiments when there are multiple structures being determined. And there's information that I'm highlighting here with the pink. That is information that would be specific for a fragment screening experiment. And then in blue here, I'm highlighting sort of the links to the other archives. And so this schematic here has sort of been broken down into these various blocks. So essentially we've got a green block here, which is essentially capturing all of that sort of information that I had earlier in an earlier slide, which is, what is this thing? What is it a protein? Is it a DNA? What it might be ligands that are, you know, a cofactor that might be present. And then also links to where various information is. And it will become a little bit more clear why this is important as I move on. So what we've done at the PDBe is essentially we've looked at what structures we have in our database, and we've found sets of coordinate files with experimental data that correspond to a fragment screening experiment. And we've found these sets of information. And then essentially we've used a software application that we've developed, and we've taken this information and generated an umbrella file, a file that sits above all of the other files, which is called an investigation file. And sort of using this little cartoon that I've generated with AI, you can see that if you've got multiple structures that are from the same fragment screening experiment, then all of the sample is going to be the same. And they'll have same alphas, potentially the same facility, and that information can all be captured in the investigation file. And so we've got a repository here where we've generated about 100 example files for this, and here's one of those example files in this repository. And as you can see, it looks similar to what I've shared before in terms of an MMSIF file. And I just want to highlight this here, which is that we are linking to multiple coordinates that are present in the PDBe file. This file is very much an umbrella file that sits on top of multiple coordinate files, multiple PDBe IDs, and interrelates those structures. And so I've just got an image here of some structural superpositions corresponding to some of the different investigation files. And obviously, once we have these kind of files, the capacity to do analysis, the capacity to do different representations, to think about perhaps generating files. There's lots of files that have partial occupancy, because sometimes fragments, you only see them partial occupancy. There might be some things we can do around that. We can really tailor a pipeline for what we do downstream once we have these kind of files, defining these structures in the context of other structures. So thinking forward, so here I've just been presenting this in the context of a fragment screening experiment, but there is, as I say, potential for this type of umbrella file to be useful for various other techniques, including time-resolved X-ray crystallography, various related cryo-electron microscopy experiments, because really the idea with these files is it enables us to link multiple IDs from the WWPDB and from other databases as well, and to link them together in order to understand multiple IDs or individual coordinates in the context of each other. And sort of as a bit of bonus content here, just included one of the ways in which this already exists at PDBe. We have the PDBe knowledge base, and essentially this is a website that you can go visit, pdbe/kb.org. If you haven't visited it for any of your structures, I encourage you to have a look. And essentially what we've done is we've taken the PDBe archive, we've done various processes, and using the fact that essentially we always map when we can to UniProt. So if there's a protein, so in this case, we've got BRCA1. It is so it's a particular gene product in human proteins that corresponds to a particular protein sequence. And we map every structure that corresponds to this to that particular UniProt ID, and then we can generate a page. And in this case, because there's some structures determined at the N terminus, some at the C terminus, you then have a page that shows both the C terminus and N terminus structures on one page. And that can be helpful. And this is another example we have here that perhaps is a little bit more relevant thinking about fragment screening, is that with this particular example, there were 413 structures solved for this particular DNA polymerase beta. And by doing structural superposition analysis on this cluster of structures, we've been able to essentially find out that in this cohort of 413 structures, they all fall within two clusters of structural, of similar structural conformer, and also we've got this other view, which is a ligand view. And if you turn sort of the clusters on and off, you can see that the presence of one ligand drives one structural conformation, and the absence of that ligand drives to another structural conformation. And this is all something that you can interact with on the PDBe KB pages. So that sort of points towards what we're trying to do as we're developing these file structures to understand structures in the context of other structures. So thank you very much for your time. I appreciate any questions, and I also need to thank the various members within the PDBe team. And I also have some, obviously, the funding bodies as well that fund our team. Thank you very much.
Thanks a lot, Genevieve, for this very, very informative talk, as usual, what you do. And I already see one of the questions in the Q&A. We have about 6 minutes. And so the first question, what here is from Vicky Hickman. Is there going to be a way to save information about a screen which isn't linked to a structure? So example for NMR screens, you won't solve a structure, but it would be nice to capture the information about hits and misses for the compound screen. Is there a database collecting this information?
So we're keen, because we're part of the WWPDB, we're already connected with BMRB. And I know that in BMRB, they're thinking about how to do this. And so it's certainly something that we're interacting with them about, that we want to evolve as well. Yeah, so it's not there yet, but it's certainly something we're thinking towards and we want to support developing, because we appreciate that just because you haven't determined a structure doesn't mean you haven't done a fragment screening experiment. That would be nice to capture in a way that can be utilized by other analysis. And yeah, I mean, the more data we can capture that we can make available in databases, the better.
Thanks a lot. I would add kind of an auxiliary for this question as an answer. Having done some screens by NMR, where did we deposit? So there was no kind of a framework. And for the initial screens, which was done on the SARS-CoV-2 proteins and SARS-CoV-2 RNA, these data were then kind of packed into zip files, and a BMRB ID was created, which means you cannot really, it's not a searchable way, but at least the whole data set is available in BMRB big. So this can be, that's the time being solution as we're trying to find more better interoperable solutions.
Absolutely. Yeah, definitely.
Okay, the second question. Thank you for the detailed presentation. Who do we get in touch with in case there are spurious cryo-EM maps and their respective PDB coordinates with wrong fold in the EMDB and PDB repositories, as they are hampering our unbiased structure determination? Interesting.
That is an interesting question. So we do have both, let me just, oh, I guess it's not there. I was just thinking the email address. So there's PDBe help. There's EMDB also has a help email that you could email. Ultimately, like we try and capture validation metrics so that people can spot when there's an issue, and hopefully reviewers can spot when there's an issue with a structure that's been deposited. But if it's been published, it's important for that data to be available for people to view, because that is associated with publication. And so, yeah, I'm assuming in this context, they're talking about a structure that's been published. That hasn't, because, yeah, unfortunately, sometimes people do deposit structures that are of lower quality / potentially even problematic. And our role in the community is to provide validation metrics so that people can understand and view them. But if they get through the review process and get published, it's important for the community to be able to view them and view that they might be problematic.
Yeah, so I do see it's the published data, Genevieve, as you also kind of anticipated. And so I guess it's most often that the best of the data, what they could get for this time, right? That's usually the game.
So you could also consider contacting the authors potentially as well, because we do have one of the nice things that's evolved over time at the WWPDB is we have the capacity to version coordinates. So if the coordinates are the problematic thing, and there's an issue with the coordinates, and the scientists who deposited it are still active in the field, they might be willing to revisit their structure. There might be something that they're willing to do. So you could contact them and ask them about it. And that is possible. If you discover there's an issue with your structure, you can contact us and we will unlock the deposition interface so you can upload a new version of the coordinates that's more appropriate for your experimental information.
Well, thanks a lot, Genevieve, and also for the audience. If you have more questions, please do type it into the Q&A, and Genevieve would be happy to answer you back, yeah, as much as possible. And now I would let the stage to Jannis, who will be speaking on leveraging the scientific language models for molecular discovery. So the stage is yours, Jannis.
Okay, thanks a lot, I think you can see the screen now. I hope, otherwise please let me know. Nice to be here. Thanks everybody for attending. So yeah, my name is Jannis. I'm an industry researcher from IBM Research. I work in the team in AI for scientific discovery. So broadly speaking, among others, we are doing a lot of things on machine learning for everything that is pre-synthesis in the drug discovery pipeline. And maybe just two preamble remarks. So first, I'm a computer scientist or computational scientist, so everything I will be presenting will be very light on sort of biological concepts and rather be focused on machine learning methods, tooling, and software. And also, so fragment to lead optimization supported by AI. So even though you might be expecting that I will be showing like lots of results of like experimentally measured binding affinities of AI generated molecules, there will be no such thing, unfortunately, because we are currently, I mean, we are running a lot of projects on this in parallel, but we don't have the final results collected yet. So I have to disappoint you on that one. So maybe just to give a real quick overview. So you've all seen the design, make, test, analyze cycle in chemistry, right? And one challenge here is that we basically, unlike in the classical scientific method, if you really think super broadly, we cannot go directly from hypothesizing to testing because we have to make the molecule first. And this, of course, is a bottleneck in this pipeline that is sort of hampering our acceleration of this cycle. At the same time, like we know that accelerating the cycle is extremely important to combat global challenges such as the climate crisis or emerging diseases. And another bottleneck is in the design step, where often hypotheses are proposed by humans in a quite ad hoc manner. And this is done because the search space is practically infinitely large, right? So you've all seen these numbers like 10 to the 60 or even more molecules in the chemical space or that are considered to be pharmaceutically relevant. The other side of that coin is also that obviously the attrition rate is extremely high, and we have only one out of roughly 10,000 synthesized molecules that makes it all the way to the market. And then the goal of our research is a little bit to sort of improve the quality of the average hypothesis that is being tested in the lab. And you can think of this as an additional small validation loop, as you see on the right here, that is simulating the outcome of the hypothesis. And we embed this validation loop into the larger cycle in order to accelerate it as a whole. So this means we are looking into molecular design. This is a generative modeling task, which means we are starting somewhere, in this case, in the chemical space, and we would like to generate new chemical matter with desired properties, of course. So we will have to produce molecules with some type of AI or machine learning model. At the same time, and obviously, of course, one aspect, and this is you see here on the right, is we would like these models to be relatively flexible. So we would like to be able to, for example, condition them on a primer or a fragment, like a substructure of a molecule that we want to start from, that we maybe want to grow or expand in the fragment growth setting. We might have other constraints that we would like to incorporate into the design, like a protein target to which we want the molecule to bind, or even like, just numerical properties like certain solubilities and so on and so forth. So we want flexible generative models. At the same time, we know that we will always have to rely also on predictive models. And these predictive models, they are indispensable for this for the task of virtual screening in order to basically nail down the most promising candidates from the virtual libraries that are produced by the generative models. So, okay, how are we going to build such models? So we will be using transformers. Transformers, you all know them from the world of ChatGPT, language models, large language models, and so on. But indeed, it's true that transformers have been applied across various domains, and the natural language is only one of them. They are very successful also in computer vision. They are used also in reinforcement learning and robotics, and of course in tools like AlphaFold. So in the biochemistry domain. So, and we will be using transformers, and transformers not operating on natural text, but on what we call like scientific languages. So these can be the language of chemistry, so SMILES, the most famous one, where you have enumerations of atoms and bonds. can be, of course, amino acid sequences, or even DNA maybe can be considered the most simple of all the scientific languages, where you have a vocabulary of only 4 tokens in the most simple case. And then we can leverage all these principles from natural language processing and train basically models on biomolecular data. So in this world, there are really lots of tasks that can be studied with machine learning. So this starts from more traditional property predictions, so predicting properties of small molecules represented as smiles. This would be classification or regression tasks. Then there are, of course, also translation tasks, like they are a bit more akin to natural text translation, where you model, for example, chemical reactions. So you go from the world of precursors, so reactants and reagents, to the world of products, or you go the other way around, which would be retrosynthesis prediction. And then there are cross-language tasks, like, for example, when you go from an amino acid sequence to a small molecule, which would be this task of like protein-driven molecule generation. So you pass an amino acid sequence to the model, and you try to produce a small molecule that will bind to it. Now, in the team, we've been using transformers across various tasks in this domain. So as you can see at the top, reaction prediction. We've also been looking into what we call paragraph to actions. So basically produce stepwise execution protocols that allow even an unexperienced researcher to perform a wet lab synthesis without having to really generate all the steps themselves. So basically, in this case, we take a reaction procedure that we fetch from a patent description or so. We take it as input, and then we produce these stepwise execution protocols. We can also do step 1 and step 2 basically together in one shot. So we arrive at the model where we can go from a reaction smiles immediately to the sequence of actions that need to be performed to conduct this reaction. Then we also have in the lab in Zurich, we have a small robotic hardware, a ChemSpeed robot that we interface basically with all these models. So, and this allows us essentially to have a fully autonomous pipeline where we can go from generative modeling over chemical reaction modeling all the way to automated wet lab synthesis. Of course, the robot is limited somehow in the types of reactions it can perform, but in general, it's supposed to be for generic organic chemistry. So all of these tools are available via this RxN for Chemistry platform. So if you go to rxn.rest.ibm.com, you will see the web page there. You can log in, create an account for free, and use all these tools for reaction modeling. And if you have the right API key, you can even launch a reaction on the robot yourself. It's just that getting this API key, you will have to get in touch with us, of course. Then I want to talk a little bit about the analogies between natural language and chemistry or chemical languages. And one thing that I think is very obvious to all of you is that language models are multitask models. So essentially, if you open ChatGPT window or any other LLM, you will assume the model is able to do text summarization or question answering or sentiment analysis, all these types of tasks. And we were wondering, actually 2 years ago, right after ChatGPT came out, we were wondering whether this also translates to chemistry. So can we use the strategy of prompted training to chemistry, where basically we build a multitask model in the chemical domain that is able to perform very really a bandwidth of tasks. And you can see here that the setup we were using for this project. So in each color is denoting one task. So as you can see, we have really text-to-text tasks. So as you can see in the top, we have a chemical reaction procedure, and on the right side, then the answer would be like a stepwise execution protocol. Here we are only looking at one of the steps. We have other tasks that are basically cross-domain. So from text to the molecule domain, this would be text-based molecule generation. So you pass a text description and you produce a smiles, or you go the other way around, which would be molecule captioning. So you provide a smiles and you would like to get a text description about the behavior of this molecule, basically. So we built one model for solving all of these tasks together, which was sort of like new because beforehand the traditional regime was just to train one model for each task. And we found actually, or one interesting observation we made was that really merging all the data across all the tasks greatly improved performance compared to only having a single task model. And even inside the model, we merged all the information. So initially we were thinking, okay, we have one encoder for the chemical data for the smiles, and we have one encoder for the natural text, but this was working much worse compared to a model that was ingesting, or like an encoder that was ingesting both together. And yeah, so we can see that especially in the cross-domain tasks that you see here in the middle, so text to molecule or molecule to text, we were outperforming the single task models. Yeah, this was a work that we published last year at ICML. It's like a machine learning conference, and I just want to highlight that these models can then be in theory. I mean, obviously this is a made-up use case, but in theory they can be useful for end-to-end discovery workflows by using a single model. So here we compared our model, so text and chemistry T5, to Galactica, which is a model from Meta, and ChatGPT, in a setting where we want to rediscover monuron. That's an herbicide. And this task involved three settings. This whole workflow involved three tasks. So first, basically go from a text description to a small molecule. So here we see that most models solved this relatively well, but only our model solved it exactly, correctly. Then the second task is once you have solved the design problem, you want to know how to make the molecule. So the second step in the workflow is reaction prediction or retrosynthesis. And here we really see that Galactica was producing a lot of garbage, whereas ChatGPT produced a reaction that was almost correct. I mean, in principle correct, just probably resulting in a very low yield. Whereas our model gave the right reaction. And then the last step would be okay, now I want to execute this reaction. So how do I do it? How do I, which steps do I have to execute? And yeah, it's not for you really to understand the details, but just to see that even here our model was capable of accomplishing this task, because obviously it was trained also on this task. So I believe that in the future, really such multimodal models, they may help us to accelerate fundamental research tasks and workflows. So this model I was just showing, and also a lot of the other work we've been doing and are currently doing, are released in this toolkit, in the generative toolkit for scientific discovery, GT4SD. So that's an open source library. Essentially, it's a Python package. You can pip install it. That is useful for hypothesis generation or for generative modeling of scientific tasks. It's a little bit focused on small molecules, but there are also generative models for polymers, for proteins, and for crystals. There are a bunch of commands that are available, like you can, for example, train your own generative model on your own data, or you can train one of the models that are already available in our toolbox on your own data. You can create inference pipelines, which means you can train the model and then use them afterwards to produce molecules, for example. You can also, if you don't want to train yourself, you can just use the models that are already there, that are already pre-trained. It works a little bit like the Hugging Face model hub. So if you would execute or run the model for the first time, it would download the artifacts and the model weights. And then you can also share and upload it to the community for others to benefit as well. We support really a bandwidth of the state-of-the-art generative models. This includes transformer models, of course, as I was mentioning, but also diffusion models, GFlow nets, variational autoencoders, reinforcement learning-based methods, and so on and so forth. Then we also have a lot of these models available on Hugging Face spaces in these web apps, which is quite neat because you can use them from a user interface without having to run any code. There will be a QR code I will be showing in the end, where you can find really, I think overall it's more than 20 models we have available there. There's documentation for each of them on how to set the hyperparameters and so on. So if you want to have a look, please feel free. And now I want to make a bit of a switch and talk briefly about 4 case studies that we are running in the context of the Fragment-Screen Consortium, which are in collaboration with 4 medicinal chemistry groups that are in Copenhagen, Madrid, Lisbon, and Riga. Each of these is focused on one protein target that is relevant in the context of SARS-CoV-2. And we are collaborating with these 4 working groups, and our responsibility is basically to take over the machine learning side and basically propose an AI-based pipeline to produce molecules that then they are experimentally testing and verifying. Now, for this, we have just open sourced a couple of weeks ago this repository that is on GitHub, GT4SD/molecular design, where really the main idea is that this entire computational pipeline that we used across all of these 4 projects, so it's pretty much the same, not exactly the same, but the main steps of this pipeline are fully open sourced now. And the idea is that even a non-computational person could bring some data. So say you have your own protein target, you're working on this, this is your PhD project, and you are interested in just executing some machine learning workflow to produce molecules that might inhibit this target, then this repo is basically for you, because without having any knowledge about programming, the idea is to just basically install an environment and run a bunch of commands, and it will basically do everything for you. Now, what happens inside, of course, this is the more important one. So there's a bunch of steps. I will walk through them now one by one. And here you just see an overview. So we already start from building a virtual screening model that is predicting binding affinity. We have a few design steps where we generate molecules, and in the end, we provide synthesis routes. So the goal here is to provide ideas for the chemists who have to synthesize the molecules on how to do that. So the first step is protein ligand binding affinity prediction. So here we use an attention-based deep learning model. And the main takeaway here is that we only need a little bit of affinity data. So this can be PIC50, KD, or even in some cases, we didn't have continuous values. We just knew, okay, we have a bunch of hits and we have a bunch of negative samples, and this is the data set. And in that case, we can also work with such data and do a classification. We importantly, and this is quite important, we do not need the target structure. So if the target structure is not available, it's not the problem. Yeah, then we train a virtual screening model on this data. Sometimes we use the data augmentation to produce or to also have a little bit more synthetic data, to have a little bit more negative data so that the models are a little bit less optimistic. Because otherwise they tend to be really overly optimistic. And then the second step is the structure design. So this is a little bit akin to fragment assembly. So here we have a lot of flexibility in this model that we're using, so we can emulate design strategies of the MedChem groups. So for example, we had one case where the MedChem group already had 2 fragments, and they knew this fragment was sort of promising, and they wanted to design a linker that can combine these 2 fragments. And at the same time, they already had done this manually, essentially, and the human expert, so to speak, had already proposed a bunch of candidate molecules. And then we were able to emulate this design strategy by running this pipeline. And we were then able to produce a bunch of molecules with this AI pipeline that were also combining these two fragments with a new linker. And quite interestingly, this I can already say, like there were some molecules that were produced by both the AI and the human expert, which is already a good initial validation. So this model here that we're using is called Molar. It's quite flexible because you can condition it on motifs, on substructures, on fragments. So there's both a way to hard constrain the molecules, but to hard constrain the models on certain substructures, but also to soft constrain it, to sort of prime it, to generate some chemical matter that is close to something that you have. But it only produces really scaffolds. And then we had to add a second design phase on top of this, which was done by another model that is the regression transformer, which comes from our own lab. And here the idea was more to do something like a fragment growth. So you start from a scaffold, but you try to expand it to improve the inhibition or other properties. Then, of course, there's a filtering stage where we take the whole virtual library and we pass it to the virtual screening model, and we only keep those molecules that are the most promising binders, so to speak. There are also some other aspects included here, like uncertainty estimation for the predicted binding affinity. And then, of course, we have to rank the molecules based on some scores. So this might be the binding affinity, but there might be a lot of other properties. This always depends on the medchem group. What are the constraints they have? They might have constraints in terms of size, like how many Daltons the molecules may weigh, the solubility, synthetic complexity. Maybe they have some building blocks that they have already available they would like to use, and so on and so forth. All of these are incorporated at this phase. And then the last phase, after we obtain a list of ranked molecules, say we converge on 5 molecules we would like to synthesize. And then the last step is the retrosynthesis search. So this is the last step, obviously not in the whole project, but on our side where we are seeing things through the machine learning lens. We are then performing this retrosynthesis where we're trying to decompose the target molecule into a set of precursors and like reactants and reagents that are commercially available so that we can produce the compound. And yeah, on that aspect, we have just published a paper in MPJ Computational Materials that shows much improved performance on multi-step retrosynthesis modeling. This is an aspect that is quite hard to capture. Like single-step retrosynthesis models are quite powerful these days, but to really capture the intuition of a chemist over a very long synthesis route that might involve like 4, 5, 6 steps is quite challenging. And on this aspect, we have just produced a new paper that, as we showed, is basically mimicking human design strategies very well. Yeah, so as I was saying, so these tools for reaction modeling, they are available on this RxN website, but also there's a Python API, so you can use the RxN for chemistry package, which is also pip installable. Just bear in mind that there's a cap on the throughput. So if you submit, say, 1,000 retrosynthesis at once, then yeah, some of these might be dangling for quite a while. But if you use it low bandwidth, it should absolutely work. Just an outlook, and then I will conclude. So what's next? So I believe the one exciting direction of for research for language models in chemistry is this idea of having chatbot interfaces that are very similar to ChatGPT, but where the backend of the chatbot has essentially access to a bunch of computational chemistry routines. So, for example, it has access to RDKit, so can generate or can execute RDKit routines, or has access to PubChem, to other databases where it can retrieve information in order to prevent things like hallucination. So you can think of a use case where you just like you paste a smiles or even a screenshot from a molecule, and you just ask the model or ask the chatbot to describe it. And then this chatbot might go to PubChem and just from there basically retrieve a description that then will be summarized for you. Then the next question you might have, okay, calculate some properties. So in this case, it might calculate some trivial properties like molecular weight, but you can have more complex ones like like synthesizability, drug likeness, and so on. And then, of course, you can make this really much more complex, that you provide a target molecule and you say, I need 3 structures that are similar to this one, but that have a log P of a certain value. So there you already see, I mean, this is a non-trivial task, right? So for this, you would actually need like either a good human expert or a generative model that can solve this. And here, then in this case, the chatbot would realize, okay, this is a design task. So for this, I have to call the generative modeling endpoint. It will provide or forward the query to this endpoint and then collect the results. And yeah, this is a little bit the direction where I can see all of this going, just to give you an outlook. And yeah, with this, I would like to close and obviously thank a lot of the collaborators who have been working with me on these projects. So yeah, happy to take some questions.
Thanks a lot, Jannis. This was really great, and also you were well on time. We have 5 minutes for Q&A. I already see the first question. What type of data do you use to train your models? Did you have to do much data curation?
Yeah, so in general, these models in general, these models are quite data hungry. But the thing was that a lot of the training can be done in an unsupervised way, pretty similar to what is being done in large language models as well, right? Where you just web scrape a lot of data and like natural text, and you train the models on like next token prediction or masked language modeling. And we're doing similar things in chemistry, but we do it with databases like PubChem, where maybe we find a few million molecules. We could even go with something like zinc, so we would have like in the range of billions. At the moment, like we mostly start in the range of like millions, but this is just for really unsupervised pre-training, just for the models to understand a little bit the language of chemistry. And there is not so much data curation being done, because we mostly just take the data as is, because oftentimes like if there's a lot of variety in the data, or like even you might call it noise, but actually to some extent is useful for the model if it's exposed, for example, to different ways of standardizing smiles, right? So there are canonical smiles, but there are also other forms of smiles, and if this is not perfectly standardized, actually for the model is helpful. So, but this is just the pre-training phase. So that of course is expensive, and this we always do it on our own hardware. Then if we talk about really use cases where you bring some data for your for the pipeline that I was just showing, so there we need much less. So it's great if we have, in terms of experimentally measured binding affinities, we have high hundreds or even low thousands. I mean, this would be amazing. But even if we have just, I don't know, a few dozens, it can already work just because the models have been pre-trained. This is at least the experience we have. So if we don't have a few dozens of data points, then it's going to become very tricky. But yeah, this is, I would say, roughly the ballpark. Yeah.
Okay, there's the second question. Is it possible that we specify what kind of structure lead that we can get eventually, for example, peptides? And also, can the pipeline work with the existed library and give some predictions?
So the pipeline, okay, let's look at the second pass first. So the pipeline, so basically the pipeline works with the library that we have been open sourcing ourselves, which is this GT4SD and also this ArcSet for chemistry. But especially GT4SD is itself depending upon a lot of state-of-the-art generative models that were produced by other researchers. And also the one model I was showing for quite a while, this Molar model, which is the first model we're using for the molecule generation, is not the model that was produced in our lab. So in general, the answer is it absolutely works also for other types of generative models. Yes. And
then
for
we have, yeah, go ahead.
So for peptides, I mean, it's not really our focus area, like protein design. But yeah, there are some models in there in the GT4SD that are for peptides, but it's more of a side exploration. I think for this, there are really better tools available than GT4SD, yeah.
Then there's a third question, Jannis. So could you tell us more about the retrosynthesis model that IBM is now using? This is the question.
Yes, so I can. So in the past, we've had successful single-step retrosynthesis models, and then there was a paper with an hypergraph exploration strategy from Philip Schwaller that was for some while the multi-step retrosynthesis model that we were using. And this is a paper from, I think, it's from some years ago, I think 2020 or 2021. And there the idea was that you represent the whole synthesis as an hypergraph, basically, where the nodes are the reactions and the edges are the the nodes are the reactants and the edges are the reactions. And this we have been using for a while, but now we have this new paper that we are using much more, where we are trying to mimic more closely the human expert design strategies. And how are we doing this? Okay, we use this concept of reaction fingerprint. So this means every single reaction is basically a point in a high-dimensional space. So it's a vector, essentially. And now, if you have a synthesis, and say the synthesis is composed of 4 or 5 steps, then you can think of the synthesis as like a time series, basically, in this reaction fingerprint space, right? Because it's basically 5 vectors in this reaction fingerprint space, but it's not just 5, it's not a set of 5 vectors, but it's more like a time series of 5 vectors, because you have to execute one reaction after the other, right? So, and what this model is doing is basically, when it's generating an artificial synthesis, is trying to align the trajectory of this of these reaction fingerprints that is proposing, is trying to align it with the reaction fingerprints of known reactions, and in this way is trying to mimic more closely designed strategies of humans. And yeah, the paper, I can just show the reference real quickly again. So this was in NPJ Computational Materials a couple of months ago, where we published this paper. Yeah.
Thanks a lot, Jannis, and we are perfectly on time, 12:01. So on behalf of Instruct, I would like to thank both the speakers for making it for the great subject and great talk, and also for all the audience for very insightful questions. And I hope we were able to give you a nice presentation here. Thanks a lot. Yeah. Unless John would like to tell something, then I would like to conclude from my side at least. Thank you.